Hello, my name is Madeleine Johansson. I work as a medical doctor at the Department of Cardiology in Malmö, Sweden, and as a clinical researcher in cardiology at Lund University. It is a great privilege to present to you our new research findings in the field of PoTS that I published last year with my colleagues, including Professor Artur Fedorovsky. I will share my screen with you. The title of my presentation today is Dysregulated Proteins in the Blood Reveals New Disease Pathways in PoTS. Here you can see the article that I published together with my colleagues in the journal Scientific Reports, where we notably identified several dysregulated proteins in the blood of patients with PoTS. I will present to you the findings and the meaning of the results today. What are proteins? Proteins are large, complex molecules that play many critical roles in the body. They are composed of long chains of amino acids that are essentially the building blocks of our cells. Amino acids are divided into two categories. There are 9 essential amino acids that can be acquired only from our diet, and 11 so-called non-essential amino acids, which are naturally produced by the human body. Our body is composed of 20% of amino acids. The function of proteins in the body. So proteins are required for the structure, function, and regulation of the body's tissues and organs. For example, to provide energy, break down food, boost the immune system, and support muscle growth, and to make hormones. The research area that focuses on studying the structure and function of proteins is called proteomics. In our study, we aimed to assess the protein background of patients with PoTS compared with healthy individuals that were matched based on age and sex. We included 130 individuals, 65 patients with PoTS, and 65 healthy controls. The mean age of patients with PoTS were 30 years and 85% were women. The mean age of healthy controls was 31 years and 80% were women. We sent blood samples for protein analyses using a powerful proteomic detection method called data-independent acquisition, which is a mass spectrometric technique generating a digital proteome, which is a map of proteins, and it's highly reproducible, and the results can show up to hundreds of proteins in the blood. On this map, you can see the different proteins that we found in the blood of patients with PoTS. And we identified in total 393 proteins in the blood, of which 19 proteins were significantly increased and upregulated in PoTS, shown in red color. And 11 proteins were significantly reduced and downregulated in PoTS, shown in blue color. And here you can see the complete names of the 19 proteins that were significantly higher in patients with PoTS compared with healthy controls. The strongest protein network interaction in PoTS were particularly associated with proteins involved in blood clotting, thrombocyte activity, inflammation, heart muscle contraction and thickening, skeletal muscle expression, and adrenergic activity. And here you can see the complete names of the 11 proteins that were significantly reduced in patients with PoTS compared with healthy controls. We observed similar protein network interactions in PoTS, with proteins particularly associated with blood clot activity, inflammation, and heart muscle contraction and thickening. So what are the findings of our study? The first important finding is that we found increased thrombocyte activity. And thrombocytes are cells that circulate in the blood, and their most important action is to stop bleeding by forming blood clots. In patients with PoTS, we observed significantly increased levels of proteins involved in thrombocyte activation. Previous reports have indicated that some PoTS patients have an increased risk of thrombosis when placing an intravenous catheter. However, there is currently limited data regarding specific complications arising from PoTS and the risk and prevalence of thromboembolism in this population. The second important finding of our study was that we identified increased inflammation in patients with PoTS. Growing evidence points toward PoTS being an autoimmune disease, often triggered by a viral or bacterial infection. In our study, we observed significantly higher levels of pro-inflammatory proteins in PoTS, such as beta-2 microglobulin, B2M, which is a protein that has been shown to be increased in patients with ongoing inflammation. Also, increased levels of this protein have been reported in several autoimmune diseases. We also noted downregulated expression of another protein called lumican, and we hypothesize that it may result in an impaired anti-inflammatory response in PoTS, with an inability to inhibit viral replication in these patients. The third important finding of our study was that we identified increased alpha-adrenergic activity in patients with PoTS. And we noted significant upregulation of proteins regulating smooth muscle activity in the blood vessel walls. And vascular smooth muscle cells are usually supplied by nerves from adrenergic receptors, in particularly alpha-1 adrenergic receptors. And they play an important role in regulating blood flow and blood pressure. In PoTS, increased sympathetic activation activates adrenergic receptors and causes a surge of noradrenaline with increased noradrenaline levels. And previous studies have also shown that approximately 89% of patients with PoTS have elevated levels of autoantibodies against alpha-1 adrenergic receptors. The second important that's the fourth important finding, is that few studies have thoroughly assessed the morphology of the heart by echocardiography in patients with PoTS. And only a small study so far have assessed the heart chambers in PoTS. And it was performed in 19 patients. And it demonstrated that PoTS patients may have smaller left ventricular mass compared with healthy controls. However, in our study, we identified increased levels of proteins involved in the heart muscle contraction and thickening. And therefore, large studies using echocardiography are needed to measure the size and thickness of the heart chambers in PoTS to see if patients with PoTS have possibly an increased thickening of the heart muscle, so-called cardiac hypertrophy. And our final important finding was that in PoTS, previous studies have shown that there is a loss of sympathetic nerve fibers in skeletal muscles, which could possibly explain why some patients in PoTS have muscle fatigability and deconditioning. In our study, we found significantly increased expression of a skeletal muscle protein. And this protein is required for proper formation and maintenance of muscle fibers and muscle function. And a faulty regulation in this protein can lead to changes characterized by a decreased muscle mass and deconditioning. So what are the clinical implications of our results? First of all, it's important to underline that few studies have identified promising biological markers in PoTS, and the therapeutic possibilities mainly focus on symptom relief, which poses substantial challenges to the physician. And our study, we identified new proteins in PoTS that may increase our understanding of possible mechanisms which may underlie PoTS. And further studies are needed to examine the underlying molecular mechanisms in PoTS in order to develop a personalized therapeutic strategy for patients. So, in conclusion, our observations are the first to use a mass spectrometry-based technique to identify dysregulated proteins in patients with PoTS. The strongest protein interactions in PoTS were notably associated with increased blood clotting activity, upregulated expression of proteins related to thrombocyte activity, enhanced inflammation, increased heart muscle contraction and thickening, increased skeletal muscle expression, and upregulated activity of receptors that bind to noradrenaline. These findings, in summary, support the hypothesis that PoTS may be an autoimmune, inflammatory, hyperadrenergic disorder. Thank you so much for listening.